Hi, everyone. I'm Julia Publicover, and I'm a Director for Translational Research and Innovation at UHN. I'm joined today by Jenny Lee and Brandon Driscoll, and today we're going to talk to you about QIPCM, UHN's Imaging Core Lab, which is also part of OICR's Adaptive Oncology Program. QIPCM stands for Quantitative Imaging and Personalized Cancer Medicine. Our core lab provides many services to ensure that multi-center studies collect top-quality imaging data and have the ability to collaborate across institutions effectively. QIPCM has established the required infrastructure and support services for international multi-center clinical trials that specialize in imaging. Our core aims of the program include a centralized data platform to anonymize and centralize imaging data across multiple institutions and allow researchers to securely access this data from anywhere. We also have a number of software tools for research use. Beyond the data platform, we have a suite of procedures and tests to ensure each study is collecting good quality standardized data. And we have a team of imaging experts to ensure everything moves smoothly for the study. So this is our team. Our PI is Dr. Brad Wouters, UHN's EVP of Science and Research, and the team consists of research associates, physicists, software developers, and business analysts. QIPCM was created just over 10 years ago now, and the number of studies that QIPCM supports has been growing steadily over the years. This graph here shows the growth over those years. The light blue line at the top of the graph is the total number of studies on the platform, which is currently just over 80. The orange line just below it is the number of unique hospitals or sites we have worked with in these studies, and that now sits at just over 65. And a couple years ago, we saw an increase in demand for support in AI studies, which is near the bottom and is in green and sits at 15. The lines labeled patients, exams, and images are all different measures of the amount of data that we've ingested, with large increases being driven by these AI studies. This diagram shows the general workflow for the platform. On the left, we have a hospital that is participating in a study, and we'll work with that site to set up our image anonymization and transfer pipeline. The tool we use is called CTP, or Clinical Trials Processor, and it's a software developed by the RSNA and is used in many clinical trials around the world. The central box is the QIPCM dedicated infrastructure, which sits within the UHN firewall. As data comes in, it first arrives in our image QC staging area, where a member of our team will perform quality assurance on the data prior to it being released to our PACs and made available to the study investigators. On the right-hand side is an investigator participating in the study. They can access the data through two-factor authentication and review or analyze the data within the QIPCM environment without having to pull the data down to their own computer. They can access the data in one of two ways: through our web viewer, which is a lightweight way to view and read images, or through a virtual machine, which may be needed by some investigators who want to do more in-depth analyses. Our PACs is partitioned so that only approved investigators can access the data for specified studies. Beyond the data platform, we have a number of services, including scanner QA and imaging protocol development. We have expertise in dosimetry for radiotheranostics, including SPECT-CT scanner calibration. And we can also support radiotherapy plan collection and remote plan review. And we have services for AI and radiomic studies as well, including data curation, data quality checks, and feature extraction for radiomics. We've taken many measures to ensure that security and privacy measures are maintained. Data is transferred through a secure pipeline, and data always remains behind the UHN firewall and cannot be downloaded to the remote computers. And two-factor authentication is always needed for login, and we always maintain a comprehensive access log. All PHI is removed at the originating site prior to its transfer to QIPCM, and our de-identification tools are fully customizable for study needs. Also, all applicable regulatory regulations are adhered to. A key security feature is that data access is only granted to individuals named by the PI or sponsor, and these individuals are only granted access to the data sets specified by the PI or sponsor. This access can be granted or retracted throughout a study as required. We have an extensive quality management system with standard operating procedures and documentation to ensure quality in everything we do is top quality and follows the applicable regulatory guidelines. Our QMS is periodically audited internally and to ensure that we are maintaining control over our processes. We've worked with many hospitals and sites globally. Here you can see a selection of our global sites, and we've worked with many, many hospitals across Canada. And with that, I'll hand it over to Jenny Lee.
Thanks, Julia. My name is Jenny. I'm a research associate at QIPCM. I'll be talking about development projects that are ongoing or already implemented on our data platform. We have two platforms. The main one is a centralized data platform, and the second one is a federated platform, which is a new initiative. This is the system diagram describing our centralized platform. As Julia mentioned, this centralized platform is currently serving many trials users, providing end-to-end medical imaging solutions. There's a lot in this slide, but I'll walk you through it. This is how we connect our researcher clinicians to the images on our centralized platform. First, at arrow number 1 is the data ingestion step. Each institute is given a CDP anonymizer, as Julia had mentioned. The trial coordinator at each site uses it to upload and de-identify images. Personal health identifiers in the metadata are de-identified and names are replaced with trial-specific subject ID. Then the de-identified data are transferred to our data repository infrastructure, shown in arrow number 2. I'll describe each component more in details later. Once data are centralized, we provide two services for our user to access the image. We could provide a VM, a virtual machine, or a DICOM web viewer, which I'll talk more later. Finally, at arrow number 4, user can log into our system using UHN as the identity provider. This step requires MFA as security measures. All data, service, hardware infrastructure as hosted and secure on UHN, represented by the yellow line. Here's some details about our data repository infrastructure. We have built it to support our day-to-day operations. The QIPCM toolbox is a web application. It acts as the single user interface for all separate tools in the repository infrastructure. Images are stored on a PAX, which is the storage system for DICOM. We use open source software called DCM Fiche, and it supports DICOM web protocol. It partitions the data by trial. Identity access management. It grants user access to the trial. We use KeyCloak as the broker and OpenID Connect for authentication and authorization protocol. We have an audit log. It records user transaction in accessing the data, when and where, what they access. Also in the repository infrastructure is image QA. It is a staging area for image arriving from external sites, where image QA are performed. We perform automatic QA for all images using Python script. Here, we implement logic to check for any remaining personal health identifier in the image or in its metadata. We also implement logic to check if images are 3D reconstructible. We also implemented modality-specific check. Example would be for CT, we need a KVP of X-ray tube currents in the DICOM tag to exist or non-empty. For radiotherapy plan, we implement logic to check all necessary components of a radiotherapy plan to exist. That is, The RT dose, RT plan, RT struct, and CT need to all be correctly linked together. We can also configure a trial to have its own custom check on DICOM tag. For example, for a trial with PET imaging, we can add a rule to check the differences between radiopharmaceutical injection time and image scan time to be a specific range. For 10% of the image, we perform manual QA on them. The QA staff go through a manual checklist, trial protocol, view images, write notes as necessary, and then pass or fail the image. To assist with the process, QA staff can customize the manual checklist and protocol for each trial from the QIPCM toolbox. Results of the automatic and manual QA are stored in a database. This is the user interface for our QA operations. On your left, shown in A, our staff can search for a trial. It lists out all studies that have finished auto QA. Then our staff can auto forward them to the final storage or select them for further manual QA. Shown in B, when QA detect an error, our staff can click on the icon to get more information about the error. Shown in C, our staff can view the results of auto QA side by side with the image. The staff can complete a manual checklist, review the trial protocol, write some notes, and then pass or fail the study. Another component in the repository infrastructure is metadata database. It stores data about the image, such as patient name, modality, study dates, and descriptions, unique identifier for the image study, results of the manual and automatic QA, or results of the radiomics feature. This database is useful to link the images to other databases, such as cBioportal. We do this by sharing partial access of this data that contain the URL of each imaging study. Brandon will describe a use case for this later. Radiomics is a set of quantitative metrics describing a region of interest on medical image. For example, some metrics quantify the shape, volume, intensity uniformity, or texture within the region. They're defined with mathematical formula and are independent of modality. They are implemented in numerical software such as Python or MATLAB. Applications of computing these metrics include predicting clinical outcome or correlating with genomics. QIPCM have implemented a pipeline to compute radiomic features using PyRadiomics. The pipeline integrated the packs with a Python compute environment together with a user interface on QIPCM toolbox. This is the user interface for our radiomics operation. Shown in A, user can search for a trial. User can search for an image set within a trial using metadata. Shown in B, user can select different settings for running PyRadiomics. Shown in C, user can optionally select radiomics to be computed on periotumoral region. Periotumoral region is the original region expanded or contracted by a margin. Once computed, the features are stored in JSON file ready for download. Once the images are centralized, we provide two ways for our user to access the image. First, we can spin up a virtual machine and give user access to it. On the VM, we can install imaging software such as MIM. We can also install development environment such as Python or MATLAB. If necessary, we can also provide GPU resource via HPC for health. The second way we provide access is through a DICOM web viewer that we host. It is based on OHIF an open source web imaging platform. It allows users to view images on the browser. It has many basic viewing features as well as tools for annotations. It renders image on the client side where pixel data are streamed to the user just like streaming movies. Data are not downloaded. Even caching of temporary files are disabled to ensure data does not leave UHN. Data are encrypted when transferred. This is a lightweight and cost-effective solution. This is a demo of the DICOM web viewer. Here, user pick an imaging study from a list and go onto segmentation mode. Then, user get ready to contour. There are different tools for contouring. Here, the user selects the standard brush tool and contour in 3D. On PET fusion mode, there are 9 viewports. The top row is for CT, axial, chrono, sagittal. The middle row is for PET, and the bottom row is the fusions of PET and CT. On your right is a MIP of the PET. Besides the centralized platform, QIPCM is also piloting a federated data platform with BCCA. A federated platform is where the imaging data can be viewed externally while the data remain at each institute. We connect the DICOM web viewer to 2 data sources, one located at QIPCM, the other at BC Cancer. The image data and infrastructure are hosted on the premises of each institute. Contrasting to the centralized model, each site takes the image, QA, and de-identify them with their own tools. Then they send it to their own data repository infrastructure, which we set up to contain the packs, identity and access management, and the toolbox as the user interface. Identity is also federated. That is, user can log in to the federated viewer with their own institute credentials and see what they're permission to see. On this pilot study, we focused on privacy and security, identity and data federations. We don't have a compute environment for machine learning yet. For user accessing the federated data, a trust relationship are in place between UHN and BCCA. That is, UHN user can authenticate using UHN credentials and then be recognized as authenticated for BCCA data, and vice versa. In this pilot, we learned that the data ingestion step could use more thought or some more tools. Decentralized and QA process and anonymization of the image could lead to inconsistency. We also learned that the system diagram, even though it's very pretty, it is much more complex in reality. Some challenges we face are staffing issue at BC Cancer and dealing with different institute IT department. Next, I'll pass on to Brandon, who will talk more about some case study for our centralized platform.
Thank you, everyone. All right. So thanks, Jenny. Good afternoon, everyone. I'm Brandon Driscoll. I'm a research associate with the QIPCM team. And I'm going to be walking us through a handful of cases which highlight how the infrastructure that Jenny and Julia have just described is applied to support a wide variety of our clinical trials and other projects. The first trial I'll discuss is called Patron. It's one of our largest trials led by Dr. Cynthia Menard out of the CHUM in Montreal. This trial is what I often refer to as one of our bread and butter trials, as it represents one of the formats that QIPCM was initially created to support. And we've done a lot of familiarity with this kind of plan. So in this case, images and RT plans from almost 800 patients across 15 sites across Canada are anonymized and collected. The images are rated locally, but also need to be reviewed by an expert central reviewer prior to randomization. This same is true for RT plans, which need to be reviewed by an expert radiation oncologist. So QIPCM supports this trial by using a Patron customized version of the CTP anonymizer to anonymize and transfer the images from the 15 sites across to QIPCM. So this uses our CTP anonymizer that Jenny and Julia mentioned, and this will anonymize the images and send them back to our image QC. We also automate emails to each of the sites upon receipt, so they know they've been received. The images undergo automated QC, checking the image completion and adherence to the imaging protocol, like Jenny discussed earlier, and then a fraction of the images are sent through manual QA, using our embedded QIPCM viewer and our QA toolbox to check for any potential burnt-in PHI and address overall image quality. Once QA is complete, the radiologist or radiation oncologist are notified there's a review pending, at which point they log into their QIPCM VM and perform that review. The QIPCM VM is just a simple Windows desktop accessed through Citrix, and it allows the users to do their review using the software choice. In this case, we can see an RT plan being reviewed in MIM. So this trial completed accrual a couple months ago, and it successfully accrued all 776 patients for the trial. And we're just storing those images now and collecting a few straggling RT plans. The next case features the work done by the QIPCM team to support dosimetry for theranostics as part of the OZM 067 NET-PRT trial led by Dr. Rebecca Wong out of UHN. This landmark trial in Ontario began in 2016 and completed accrual in 2021 and helped pave the way for Lutathera to become a funded treatment in Ontario today. Before I get into this trial and how we supported it, I'd like to say a few words about theranostics in general. So theranostic agents are an exciting new area in cancer treatment, where a specific targeted agent can be utilized to image a patient, while a companion version of the same drug can be used to then treat the patient with a different radioisotope. So in this case of lutetium dotatate, dotatate binds to a somatostatin receptor, which is overexpressed in many tumors, but primarily neuroendocrine tumors. If you bind dotatate to gallium-68, you can perform a PET-CT of the patient and determine the extent of disease, as you can see on the left here. But then you can add a lutetium-177 isotope to treat the cancer with the radiation from its emitted beta particles, while imaging it using the emitted gamma rays via SPECT. This allows us not only to treat the patient, but know quite precisely where and how much we're treating. The OZM 067 trial planned to study 190 patients from 4 sites across Ontario, and each patient would receive that gallium-68 PET scan to determine eligibility, followed by 4 cycles of dotatate treatment, while having personalized dosimetry performed at each cycle via SPECT-CT. So as in the previous trial, we started off by setting up all the sites with the trial-specific version of the CTP client. This allowed us to anonymize and transfer all the images centrally to QIPCM. Our team then supported performing SPECT-CT sensitivity QA at each of the 4 sites to ensure we could perform quantitative dosimetry. We then performed QA on all the images upon receipt and notified the research therapist that images were ready for segmentation and subsequent dosimetry. So QIPCM worked with our colleagues in the RMP physics program to develop an individualized dosimetry extension using a combination of MIM and MATLAB. This workflow provides an automated image QA procedure, which allows for the generation of personalized dosimetry reports, which track the dose to both the organs of risk as well as tumors across all 4 cycles of therapy. So you can see an example of that process here, where we would use MIM to do the segmentation, and then the back-end dosimetry is computed in MATLAB, and our reports spit out for sign-off by the the medical physicist and the radiation oncologist. Now, after successfully completing the OZM 67 trial, we've been working to use the same dosimetry process for the standard of care Lutathera treatments at UHN. So the third case I'd like to talk about, we're going to switch streams a bit here to the Octane trial led by Dr. Phil Bedard out of UHN. It's a good example of how QIPCM worked with other teams to linking our database with their clinical data. As in the previous cases, both Octane 1 and 2 will be collecting large amounts of imaging data from sites all across the province and centralizing it here at QIPCM. However, the primary difference with the Octane trials is that they're also collecting a large amount of genomic, and tissue sample data, which will be made available via another web-based solution called cBioportal. The hope was that we could work with the cBioportal team to include the imaging data along with the genomic data for these patients. The CTP anonymizer was again set up to anonymize all this data and centralize it to QIPCM, where it underwent automated manual QA by our team using the QIPCM imaging toolbox. We then worked with the cBioportal team to create a link between their platform and our viewer to allow study researchers to view imaging data alongside their other clinical data. Additionally, as part of the trial, there was a plan to extract radiomic features for these imaging sets, and that data could also be made available in cBioportal. So here you can see the cBioportal dashboard. And if you click on a patient, it'll bring up their individual patient dashboard. And in this dashboard, it shows their imaging. You can see each of these gray time points is the time point at which the patient had imaging. And there's a link directly to the QIPCM DICOM viewer. I don't know if you noticed, but the ones in green there are the ones that are highlighted that have RT structs in them. So these RT structs are great because we can perform radiomic extraction on them. And you can see the link was quite straightforward. It launched right into the QIPCM web viewer, which then can load up and show us those images and show the RT structs and where they're located in the patients. So this fourth case involves using the QIPCM platform for educational purposes, which is something new to QIPCM that we hadn't really explored before. And in this platform, the goal was to create a database that could be used to train radiologists and radiation oncologists about the best ways to read and interpret PET PSMA images. This project's led by Dr. Catherine Zukatinsky using PET PSMA registry data obtained from Dr. Glenn Bauman at London Health Sciences Center. So Dr. Zukatinsky came to us looking for a way to create and host a database of PSMA PET images, complete with annotations and case reports. She wanted this information to be made easily available via a web viewer to other radiologists and radiation oncologists to train them on how to better read and understand novel PSMA PET images. Dr. Zukatinsky also hoped that this curated and annotated database could be utilized to train machine learning algorithms to better detect PSMA-avid lesions. So QIPCM worked with Dr. Zukatinsky as well as representatives from the London Health Science Center and our own Grants and Contracts Office to ensure there was proper data sharing agreements in place for the trial. In this case, they were a little more complicated than normal because they were being shared between 3 different parties, and then the hope was that the data could be shared visually, not allowing download to the learners using the platform. We then worked with Dr. Bauman at LHSC to collect, anonymize, and curate the data such that it could be annotated for the study. To do this, we provided a virtual machine to an expert nuclear medicine radiologist to use to annotate and segment the imaging, as well as create all the case reports. Here on the right, you can see an example of one of these case reports alongside the segmentation done by the expert nuclear medicine radiologist. And this is being shown in our OHIF viewer, the QIPCM OHIF web viewer, that'll be used for the education users. We're currently in the process of working with Dr. Zukatinsky to create a set of YouTube videos to help train learners to properly use the QIPCM viewer and navigate the education cases, as well as accurately review the PET PSMA cases. And discussions also continue with our collaborators looking at ways to expand this annotated database, as well as the best ways to use it for AI training. So the final case study I'll discuss today involves supporting the development of AI-based tools and models by supporting our industry partners through the use of the QIPCM AI Sandbox. So there are many industry partners who would like to build and train AI and machine learning algorithms on UHN's valuable imaging data. Unfortunately, questions of data ownership, patient privacy, and intellectual property concerns make it virtually impossible to export UHN's clinical data to industry partners directly and to develop new tools so they can develop new tools to better detect and treat cancer. Our solution has been to anonymize and collect the data from UHN's clinical packs and provide it to the researchers on the QIPCM framework, so the industry partners can leverage the anonymized data without it ever leaving the hospital. In order to make this happen, QIPCM works with a UHN-based primary investigator, as well as the JDMI AI office, or the JDMI Artificial Intelligence office, to link industry partners with the appropriate patients and clinical data. The industry partner and PI will develop a clinical question, and then they'll work with the JDMI AI department and other clinical departments within UHN to find lists of patients and MRNs and associated clinical data who fit their criteria. Those lists are then provided to QIPCM, who merge them into a master list and use that to query the UHN packs and determine what imaging is available for each of these patients. That information is anonymized and given to the AI team, and then the set of images is retrieved from the packs, anonymized, QA'd, and then provided to the industry partners on the QIPCM AI sandbox. So we refer to these virtual machines as the QIPCM AI sandbox because they're segregated from the rest of the UHN network based on the user's access rights. These external users have access to their QIPCM virtual machine and QIPCM hosted shared drives without having access to the rest of the UHN domain. Since the QIPCM virtual machines are simply Windows desktops, we can provide a wide range of different AI development tools on them, such as Anaconda, Python, 3D Slicer, Git, R, MATLAB, etc. And finally, if our industry partners need additional computational power, QIPCM VMs are connected directly to UHN's HPC for Health, which is high performance computing for health computational cluster. And this can be used to queue up large volume machine learning and model building experiments. So hopefully you found each of these case studies interesting and learned a bit about how QIPCM supports their research projects. And with that, hopefully we've got plenty of time left for questions. Thanks, everyone.
So thanks so much, everyone. We hope you learned a little bit about what we do. I can see there's a couple of questions in the chat, so maybe I'll read them off and either I can answer them or I can throw them to either Brandon or Jenny. So the first one I see here is, can a researcher work directly with de-identified DICOM data? Brandon, do you want to take the first crack at that one?
Sure. I mean, I guess that one kind of depends on the context of the question, but yes, researchers can work directly on their own de-identified DICOM data. We don't have a repository that's open for sharing for just anyone. So the data that's on here is hosted for the PI and only for the PI. Now, if they want to share that with another investigator or another research team, that's completely fine. But it's not like a large data repository that other people can use. But if it's their own data, yeah, they can come on, they can use the tools from the VMs, or they can just view them through the web viewer.
No, but can they actually download the data, for example, because there might be debugging Slicer or doing some type of work like that, for example, or does the data have to remain within your side?
The data does remain within the QIPCM platform. So our model has always been that we basically create that's what the virtual desktops are for. So if they want to do their analysis, we provide them with that virtual desktop with access to Slicer. They can put their code on there and they can run their tools there. We don't tend to allow, there's no way for the user to download the data off themselves. If the trial wants to just get us to collect everything, and at the end we just extract it all, embed it on a hard drive, an encrypted drive, and send it to them, that's another model we can do.
Thank you.
Thank you. So the next question was, what kind of resources and support does QIPCM provide to researchers from other countries, and how can they get started with accessing these services? Maybe I'll start, and then Brandon, you can follow up if I missed anything, or Jenny. So we can provide almost all of the same services to researchers in another country that we do here in Ontario and in Canada. So we are part of the OICR imaging program as well. So our mandate is really to help research sort of across Canada and globally as well. We can help a PI who's located in another center in another country and centralize the data for them. We can also provide access to researchers in another country who might want to log in and be part of the research team. Brandon, did you want to follow up with anything? We do have researchers from outside of the country who both, collected their data on the platform as well as been part of the analysis team.
Yeah, I think that generally covers it. I mean, the one important feature, I guess, would be just in terms of regulations and where different people might be hosting their data. So there might be different regulatory requirements based on that. But in terms of actually using the services and how it's provided, it doesn't really matter. We've had people access it from everywhere.
So the next question is, what are the key considerations for data security and regulatory compliance? Jenny, do you want to talk about sort of what we have technically for security and regulatory compliance, and then maybe myself can address what we have for processes?
Right, so all data are hosted within UHN. That is, our services, our infrastructures, and our data storage are within UHN. For user to access the data either through a DICOM web viewer or via VM, they would need MFA, multi-factor authentications, and that is using UHN as the identity provider. So in that sense, that's our security measurements that are in place. And even when we use the DICOM web viewer transfer to the DICOM web viewer or the transfer through the web are all encrypted.
Thanks, Jenny. Yeah, so since at the very beginning, we knew that security and privacy and regulatory aspects of this were going to be very, very important. And so we built the infrastructure in order to assure all of those check marks are made. And we have all the processes in place to ensure that we are following all of the applicable guidelines for security and privacy as well. And we consult with UHN security and UHN privacy regularly if we're going to implement something new. All right, and the next question. What specific benefits does the QIPCM AI sandbox offer for AI-related medical imaging research? Jenny, do you want to take that one?
Yep. So some of the benefits of the AI sandbox are that the user will be given a virtual machines, which is essentially like a Windows desktop environment and access to a shared drive. And on the Windows desktop machine, we can install any software necessary, such as Python, MATLAB, 3D Slicer, anything that the user are familiar with. And for machine learning, when they need more compute power, we can give access to GPU resource via HTPC for health. So the benefits there is that you're familiar with the environment, which is just like a Windows desktop, and you can install any software you need and have access to GPU and large storage. Brandon, if you have anything else to add?
I think the only other thing I'd add is just the alternative to a lot of this would be companies pulling this down and doing their research on the cloud, and it's very challenging to do that with data that's coming out of UHN. So this allows us to keep that anonymized data within UHN, so that it's still contained within our firewall and under our general control.
Great. So we got another question that came through from Michael. How does QIPCM connect electronic medical records data with imaging data? How do the users then interact with the combined data set? Brandon, you probably have the most experience with this.
Yeah, ultimately, QIPCM for years has just dealt with the imaging data. That's been our focus. When I presented the stuff about Octane, that's been one of our first kind of forays into dealing with the clinical data and linking to it. We don't have any of that access to that. We don't even use Epic or EPR. That's not something we even have access to. But we do work with the other people like cBioportal that already do get a lot of that information. Subsequently, we also have worked with AI companies with industry partners who are trying to pull in both. So we'll work with the JDMI office and PIs to get that data from the clinical departments and merge it together based on their IDs. And then it just comes down to anonymizing the data before it goes out to the clients. The one big challenge there still is getting full like case reports and that kind of thing. We haven't really touched that yet. We're looking at, we're not specifically, but there are people at UHN looking at doing natural language processing and that kind of work to try to pull out the information from case reports. But ultimately, we're just looking at tabular reports like biomarker stage, follow-up, or not follow-up, but like outcomes and that kind of thing. But yeah, we don't really house that database. We'll just work with the investigators to kind of merge it all after the fact.
Great, thank you. Oh, and can users tap into larger compute resources like what is available at the Digital Research Alliance? Jenny, you want to take that one?
So we're not affiliated with Digital Research Alliance. So the short answer is no, but we are familiar with them. We know generally what they are doing. I know they're making big progress in trying to share digital cloud resource. So the compute resource that we are offering that we can do at the moment is a HPC for Health for GPU access. So that's what we can do right now.
All right. Excellent. Well, thank you so much, everyone.
Thanks. Bye.
Bye.